Today we'll discuss about the identification of gram-positive bacterial isolate. So although the identification of gram-positive bacteria is not very wide, so still we should know what are the basic biochemicals which are required to identify these organisms. So gram-positive bacteria, once we make a smear from a particular lab sample, we can identify whether it is gram-positive cocci in chains or gram-positive cocci in clusters. So when there are chains present in a sample, because of gram stain, we can identify them as gram-positive. That will appear as violet in color. So gram-positive chains will grow as Streptococcus species or Streptococcaceae, and the cluster will grow as Staphylococcus species or Micrococcaceae family. Further, for identification of Streptococcus or Enterococcus, we will further put up a set of biochemicals. First, when we see the growth on a particular plate, we pick up the colony and we will perform a catalase. So the organisms belonging to Streptococcaceae or Enterococcaceae, it will be catalase negative, and then we'll further proceed by putting up the bile esculin agar, that is BEA, 6.5% NaCl or bile esculin. So these are the basic set of biochemicals which are required to identify the Streptococcus. So once we perform catalase from the nutrient agar plate, and if the reaction is positive, that is catalase positive, then we suspect the organism to be Staphylococcus or Micrococcus. So further, we'll put up a certain set of biochemicals to differentiate the organism further. So the important test is coagulase, which can be a slide coagulase, tube coagulase. There's a DNase test which is put up. Then there's an important media which is used, that is mannitol salt agar, which is specific for Staphylococcus aureus. The Staphylococcus aureus, it will give a yellow colored colony on this particular agar. Then another important identification method is putting up the AST,